Hello to you all awesome matriculants. Happy Wednesday. I hope you enjoyed your day at school and you are studying, but thank you for joining us. My name is Abram and I'm joined by Mr. Aslam. It's the A squared team.
A squared.
Yes.
A cube.
Yeah,
A squared. Watch it. I don't know anymore.
It's an A squared.
We need to find a catch line for Wednesday. So let's throw it out to the mindsetters and tell them they must come back to us. What will we call our life science Wednesdays? If the best guy gives us, we're going to use it every Wednesday.
Right, guys, you have a challenge. Send that to us, your suggestion on how we should call our life science on Wednesday with Aslam and Abram. Otherwise, what are we doing today?
Today we're talking about meiosis. Meiosis.
Oh, meiosis.
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 Yeah.
All right, my sisters, if you have some questions, feel free to throw them to me on Facebook at facebook.com/learnextra. Like the page and share the page with all your friends. Tell them your teachers also at school, share to them about mindset. Also, follow us on Twitter at learnextra. I'll read your tweets and see what are you guys saying on Twitter. Otherwise, get your notes all for free at learn.mindset.co.za. Now, on your notes, you have your test yourself questions, whereby after you've downloaded the notes, write down your answers and then submit them to us on the link that I'll post on Facebook. The winner will be announced tomorrow because we have a competition whereby if you get all the answers correct, you'll stand a chance of winning an airtime voucher proudly sponsored by Vodacom. So get your answers coming in on the link that is posted on Facebook. But for now, you'll be telling us about the challenge question also.
Yep. Quite a bit of stuff that we're going to do. We're still going to first talk about what are we doing today. Today we're talking about all the phases of meiosis 1 and meiosis 2. Remember that meiosis has 2 divisions. State the importance of meiosis. In other words, why does meiosis take place? State the state what is meant by non-disjunction. Non-disjunction. Non-disjunction, a junction is a joint normally. Disjunction means separate. Non-disjunction means when something does not separate. We'll talk about that later. State the consequences of non-disjunctions. In other words, first we're talking about what is non-disjunction, and secondly we're going to talk about what happens if non-disjunction does occur in meiosis. We also want to talk about how this non-disjunction of chromosome pair number 21 leads to Down syndrome. Okay? Describe how the fusion between the abnormal gamete and normal gamete may lead to Down syndrome. There, at the end there. We also want to know what are the similarities between mitosis and meiosis. And lastly, we want to know the differences between mitosis and meiosis. So this is what we are looking at today, guys. Very simple. We're looking at meiosis, first of all, and want to explain what meiosis is, what happens, what's the significance. We also want to talk about, in certain cases, what can go wrong in meiosis, what's the problem, and what can, specifically in our case, we're looking at one example now, and that is Down syndrome. There are many other syndromes that we can talk about, but the syllabus prescribes Down's syndrome. So that's the one we're going to concentrate on. If in an exam they ask you any of the other syndromes, they must give you enough information in the passage before the questions so you can understand it. They may just test your understanding of meiosis. And lastly, we must also look at mitosis and how is it similar to and how different it is from meiosis. Good. Remember you did mitosis in grade 10. Your challenge question, AB was jumping for the challenge question. Just to read it for you quickly. If X units of DNA are present in the nucleus of a cell, which has just divided, what is the relative amount of DNA present in this cell during prophase of the next mitosis? In other words, this cell has just divided. Now, the new daughter cells, they want to go to and also divide. Let's say it's your skin cells or your nail cells or whatever, because we're talking of mitosis, remember? And that's the question there. Okay, maybe I've given you a little bit too much in that, but nonetheless. The second question I like more, if a rhino consists of cells where 2N, and you should know by now that 2N is the diploid chromosome number, equals to 82. How many chromosomes would it have? A, in the cells of its ears, in the cells of its horn, in the sperm cells, in the cells during anaphase 2 of meiosis, and in the cells during prophase 1 of meiosis. Some thinking you need. Put on your thinking caps and think carefully about this question. And let me tell you something, guys. If you can answer this question correctly, that means you have grasped this concept of meiosis properly, because meiosis is all about this. Good. That's the question. Keep your answers coming on the page, and we'll take it from there. Let's look at the summary of what we are talking about in this section. Again, we're talking about meiosis. We talk of the first division, where there is prophase 1, metaphase 1, anaphase 1, telophase 1. And in between, before that division, before the other one, there will always be interphase. All right, and then we go into division 2, where it moves from telophase to prophase 2, metaphase 2, anaphase 2, telophase 2. Notice the names of the phases have not changed from mitosis. In mitosis, you learned the phases. They did not change now. They're still the same. And if we take the first letters of each of those phases, this is what we will get. I, P, M, A, T. So you can make your own mnemonic there. I prefer the mnemonic. Wait for it. I pee on the mat. Not a nice thing to do, Abraham. Not a nice thing to do, to pee on the mat.
Nope.
But to remember this, I pee on the mat. I for interphase, P for prophase, M for metaphase, A for anaphase, and T for telophase. I've got one of my fans laughing, so I'm hoping the rest of you also are giggling with that, what I just spoke about. So remember that that's the order in which the phases unfold. Remember this year, you need to know the names of the phases. Last year's matrics, up to last year, from 2011 up to 2013, the matriculants did not need to know the names of the phases. It was ridiculous anyway, but now we've corrected that. You need to know the names of the phases and what happens in each of those phases. So you need to describe. You need to describe what happens in each of those phases. All right? Thereafter, you must also talk about the importance of meiosis. In other words, another word, the significance, or why. Why does meiosis take place? What's so good about it? And the consequences of abnormal meiosis leading to Down syndrome. And you must be able to compare mitosis and meiosis. Good. We're now going to look at a small little snip on meiosis to understand what is happening in this particular process. Okay? So you need to watch carefully. Wait.
Okay, while you're trying to fix that, let me tell the mindsets apart.
Tell them that, please.
Right, Mindsets, you have the challenge question posted on our Facebook page, which is facebook.com/LearnExtra, and follow us also on Twitter @LearnExtra. If you're having any questions, feel free to throw them to us. We'll answer them later on the show. I see there are already some Mindsets asking on the Facebook page, and a big shout out to Letuk Tula saying, "Hi AP, please post the link to the challenge question. It's already posted. Please check it out." And to other Mindsets saying they're looking forward to today's lesson, like Esite Yonji. Looking forward to today's lesson. Keep your questions coming with Aslam.
All right, there we go. We got our meiosis DVD or playing here for us. I want you to watch carefully, but I'm going to guide you through it. This first part is just showing you an ovum and a sperm coming together. Now, what I want you to do is watch here what we're saying. We're saying that if a sperm has 46 chromosomes and an egg has chromosomes 46, then the resultant zygote, 46AB plus 46, will give us
92.
92. Okay? So in other words, what we're saying here, guys, if an egg had 46 and a chromosome, remember why we're using the number 46? Because humans have 46 chromosomes in their ordinary body cells. All right? Somatic cells, ordinary body cells, the somatic cells. Now, if that happens, then we're going to have problems, okay? Now, here they're telling you about the baby. We're not worried too much about that. There we go. Now what happens? We're saying that because of meiosis, the sperm has 23 and the egg has 23. So what has happened? We have reduced the chromosome number by half. Why? So that when these 23 chromosomes join with these 23 chromosomes from the sperm to the egg, what are we going to get now, AB? We're going to get 46. Now let's understand this concept, because this is the crux of meiosis. This is why, one of the main reasons why meiosis takes place. Let's take that we are all descendants from Adam and Eve. So Adam and Eve then would have had 46 chromosomes each, and their children would have had 92 if meiosis did not take place. And their grandchildren then would have 184. And so we would have gone on 368, and so on and so on and so on. The thing about chromosome number is that it is specific to the species. Chromosome number is unique to a species. For example, we have 46 chromosomes. Ferns, on the other hand, some ferns have 300 AB, 300 chromosomes. So the chromosome number is not an indication of modification or more smart. Can you see? The fern, which we humans think are lower order, and they are plants. And we, on the other hand, have 46 chromosomes only. So that's what we need to think about there. Okay? Now let's look at what happens during meiosis. And we're going to go through the phases very quickly. This is showing you prophase. Watch what's happening. The chromosomes, just watch as it happens. Remember, in interphase first, the chromosomes are in single strands. Watch what happens. What happens? DNA replication takes place, and the chromosomes are now formed fully. And that's what we call prophase. And the highlight of prophase is that the chromosomes become visible, and more importantly, the homologous chromosomes lying next to each other. Remember, in mitosis, all these things happen. The nuclear membrane disintegrates, the nucleolus disintegrates, the spindle fiber forms, and the chromosomes become visible. Mitosis and meiosis. But in meiosis, the homologous chromosomes lying next to each other. And as we proceed to the next phase, which is metaphase 1, the homologous chromosomes lie in the middle of the cell, at the equator. Okay? But notice that homologous chromosomes are lying next to each other. Don't be confused with the color. Look at the size of the chromosomes here. The longer chromosomes are lying next to each other, and the shorter ones are lying next to each other. So this one here on the left is a homologue of the one on the right. In other words, one of these chromosomes you got from your mother, and one you got from your father. Okay? During anaphase 1, what will happen is the chromosomes move to opposite poles. Notice we're not saying the chromatids, we're not saying that the centromere broke, nothing like that. We simply say chromosomes. The whole chromosome moved to opposite poles. And during telophase, these chromosomes will be at the poles, and the nuclear membrane will reform, and immediately we go into prophase 2. There you see the chromosomes in prophase 2. And as we move on, you'll notice that the chromosomes start lining up at the equator. Now look, in this particular cell, and I'm glad that they're showing it this way. The other one, we had the middle from left to right. In this cell, they're showing you middle from top to bottom. In a cell, it doesn't matter whether it's side to side or top to bottom. The middle is considered to be the equator. It all depends how the spindles form. So here you have your chromosomes in the middle here. But how are they now? Are the homologous chromosomes lying next to each other? No. The homologous chromosomes are lying independently now. For a start, they've been separated. So each chromosome is lying independently at the equator. Very similar to mitosis. Okay? And what will happen then? The centromeres will break, and watch what happens as we play on. There we go. So where we have anaphase 2, what has happened now? The centromere has split, the spindles have contracted and starting to pull these chromatids apart. Remember, I emphasized in anaphase 1, chromosomes move apart. In anaphase 2, chromatids move apart. Okay? And moving towards telophase, the chromatids are arranged at the equator and at the poles, and as you can see, we have now 4 cells with half the chromosome number as the original cell. Original cell had 4 chromosomes. Each of these cells have 2 chromosomes. We can see that clearly in this diagram. And as we're moving on, we will see that the cells will undergo interphase and they will then make their story, and there's your sperm and ovum fusing, and that is where we would stop that in terms of our summary. Remember, the idea of showing this video was to show you physically how the things move apart. Like a movie. Some of you watch a movie once, but after 10 years we will ask you about that movie. You'll be able to tell us what happened at which time. So in the same way, we want you to remember this movie called Meiosis, with A squared showed you this movie called Meiosis. We want you to remember it. Ebi.
Awesome. We're the proud directors, right?
There we go.
All right, Meiosis, keep your questions coming. We'll see you after the break. Welcome back, Meiosis. Now, hearing the good news that some of you, like Khutso, say, I always wanted to watch that Meiosis movie. Now I'm satisfied you enjoyed our movie. How about this? I'll post the link of the movie to our Facebook page. So get over Facebook, facebook.com/learnextra, and check out the link that I'll post for the movie. Isn't that great?
That's great, Abi. At the same time, just remember, Mindsetters, there are plenty movies like this. Once you go on to Google and you just type in Meiosis plus video, you'll find 1,000, right? Some are not great. You'll have to filter and go through and see that we've selected what we think would be easiest for you to understand. All right. Let's move on, guys. We're now at that segment of the day, which is the most important. Remember the first part, you've already done that work at school. We are just recapping what you've done in work. But this segment is about, the next 2 segments are very important in that we are taking exam type questions and going through them. At the same time, you posting your questions, which we're going to take in the last segment, and we're going to get the challenge question going as well at the end. Let's start with question 1. Study the diagram and answer the questions that follow. Very simple instruction there, nothing elaborate about the instruction, but they're giving you a diagram here and they're showing you something happening there. And A, B. Okay. So the first question says, what type of cell division is shown here? What type of cell division is shown here? Okay, in today's time, it's easy because we're doing meiosis. It's easy to answer that question as meiosis. But remember, there's also mitosis that can be tested in an exam. So we are saying what type of cell division taking place there. It's obvious, it's meiosis. Now, obvious as well is the minute they ask you what is this thing here, the next question they're going to ask you is provide observable reasons for your answer. And notice the observable, although it's spelled incorrectly, but it should be observable with an R there, observable reason for your answer, okay? Although it's spelled incorrectly, but it is in uppercase, normally bold even in your exam paper sometimes. Why? Because they're trying to get to your attention that, listen, guy, we know you know lots of other reasons why this could be a meiosis. We're not interested in those reasons. We are only interested in what you can see in this diagram. And in this diagram, what can we see? One, we can see that the homologous chromosomes are lying next to each other, one. And two, we are also seeing this important process here called crossing over. All right, so there we go. Homologous chromosomes are lying next to each other and crossing over is taking place. Any one of the two is an acceptable reason. Now they want to know, rest, you're telling us it's meiosis, very good, you're very clever. Now tell us which phase is it? What is the name of the phase here? And obviously, because of what we said here above, it has to be, according to what we discussed earlier, it has to be prophase 1, because only during prophase 1 do the homologous chromosomes pair up like that and crossing over takes place. Now let's explain what crossing over means very quickly. Crossing over means that the chromosomes that are lying next to each other, this chromosome, you got one from the mother, one from the father, maternal origin, paternal origin, the homologous chromosomes. Each of these two chromosomes code for exactly the same thing. If the one is coding for eye color, size of nose, size of bum, size of finger, whatever else that it could be coding for, the other one must code for the same thing, because you got this set of characteristics from your mother and this set of characteristics from your father. So that's what happens. But when they cross over, what happens? Parts of the one are switched over to parts of the other one. So when this crossing over is taking place, these chromosomes are not as they were before. And this brings about variety in species. That's why you do not look exactly as your brother, AB, or your sister, unless you are
Twins.
identical twins. This is the variety that we're talking about. Otherwise, we would have all looked like Adam and Eve. Imagine this scenario, AB. Adam and Eve and all their children were all the girls were looking like Eve and all the sons were looking like Adam. And in today's time, imagine that was still the same. I would come to you, AB, hi Adam, how are you? And you would say back,
Hi, Adam, I'm good.
okay, and Adam, how is your wife Eve? And what would you say?
How was your wife to Eve?
Like, there we go. And imagine I'm the cop and you the criminal. And I say, hey, Mr. Adam, can I get your fingerprints? What can you tell me?
No, Mr. Adam, you can't get my fingerprints.
Why?
Because you're Adam,
Exactly, and my fingerprints would be?
like yours.
So there you go. You can't catch any criminals. So imagine the world without all this. Now imagine that all this was put into Adam and Eve before, AB, this laptop was invented. Before Google. Before Google.
Our
In
best
today's
friend.
generation, if you ask them what was life before Google, they will say, ish. What can we do without Google? Eh? You want to write a speech? Google. You want to find a mate? Google. Eh? The only thing they can't do, we'll talk about next week. What they can't do on Google. But anyway, even that today you can.
You want to find a Meiosis video?
Google it. There we go. Right. Okay. So imagine the beauty of creation, people. These are moments, while we laughing about it, these are moments that make us think about, that has to be a creator that put all this, has to be some higher power to put all these things without Google. Huh? Okay, what importance process is shown at B? We've discussed that in detail already, crossing over. And what is the significance of the above mentioned process? I told you that already, and that is, it allows for the exchange. Listen to this question and listen to this answer, and I want you to register it. Allows for the exchange of genetic material. And this brings about variety or variation in species. In other words, it makes us different. It makes plants different from each other. All species that are not exactly the same. And this is important because variety means that if there was an ailment and if this ailment had to be in all the offspring, then all the offspring would have that ailment and they would be weak. Where when we get variety coming in, this now increases the chance of survival within species. Remember, it doesn't mean that you cannot get an ailment because of the variety. It just means that you stand a better chance. If the two partners that resulted in you being born did not both have this characteristic that brings about this ailment. Okay? Let's move on. Draw a fully labeled diagram to show the phase occurring immediately after this phase. So let's go next to this one here, then we know what we're talking about. For a start, what are we talking about? We are talking about metaphase 1. So obviously we will still have the cell membrane. Remember in the exams, when they draw a membrane on the out, it's not the nuclear membrane because the nuclear membrane disintegrates during prophase 1. Okay? So this will be the, let's label it while we're there. Cell membrane. Huh? Okay? Then we'll take these chromosomes. I'm not going to draw them exactly like they are there. I'm going to draw my own chromosomes. And just to show a little bit difference, so what we are showing here is that these are the cell membranes, and then we can put a curly bracket there and say homologous chromosomes together. And if you draw in this diagram, put this like this, together at the equator. So when you draw in this diagram, you will know exactly what this phase is. And guys, the best way of learning meiosis or mitosis or any process is by drawing the diagrams yourself. Draw your own diagrams and draw annotated diagram. Annotated diagram means draw a diagram with notes, meaning if you're talking about it, say a sentence or two, what is happening there. And if you draw this on a double page, take your book, open a double page, and draw the phases of meiosis 1 and the phases of meiosis, or you can do it on a separate double, the meiosis 2, and each one label the diagram. Now don't label chromosome every time, because you've labeled it once, you know what is a chromosome. As you're going on, you're going to label what you see new. More importantly is what you see happening more, or new, sorry. And this series of pictures must tell you a story. So when you're remembering it in an exam, you're remembering pictures, not whole chunks of text, and you explain the pictures you see in your mind's eye. So that's the because this is a diagram we're drawing, we must have a caption. And here, diagram showing metaphase 1 of meiosis. That's what we have to do there. Got it? Great. Okay, so that will be the diagram you're drawing. Good. Let's move to our next question. They're saying here, bacteria reproduce asexually by binary fission. Important note there, not sexually but asexually. And this is by ordinary mitotic division. 2 sterile petri dishes are filled with neutron agar, etc. You got this in your note. I'm not going to read the whole question. All right? Provide a hypothesis. Now remember, although the bacteria is not part of the syllabus in this particular section, however, knowing how to make a hypothesis is. And once you learn the skill of a hypothesis, you'll be able to do that. So provide a hypothesis for this experiment. And that would be, number 1, either ultraviolet light causes mutation or does not cause mutations in bacteria. You don't write both, obviously, in your answer. You're going to write one, because this is a hypothesis. You could have hypothesized that it does, or you could have hypothesized that it does not. You'll have one of those two. Or you could say that bacteria mutate or do not mutate in darkness, okay, because of the variables that were there. The important thing is you must have the independent variable. In this case, what can you change? You can change the light. So that's the independent variable. The dependent variable is a mutation, whether the mutation is going to take place or not. And the relationship is this word causes or does not cause. You cannot put the hypothesis in a form of a question. Will ultraviolet cause mutation? That's an aim. That's why you're doing an experiment. That's not what. You must take the chance. You must say it will or it will not. So you must show a relationship. And maybe the best example we've done before, last year or the year before even, we spoke about a hypothesis. You must have one hypothesis in your head, which you use as a template, and you fit in the new information in there. The one I like to use, AB, is, if I give her a diamond ring, she will marry me. Let's write that down.
I'm going to steal that one.
Let's write that down. If I give her a diamond ring, I'm not writing it full, she will marry me. Now, AB, which part are you in control here? Her marrying you or giving the diamond ring? Which part you in control of?
Giving of the ring.
Giving of the ring. So giving the ring is what you, the investigator, can do. So that becomes the independent variable. It can stand on its own. You can either give the ring or you don't give the ring. But whether she marries you or not It's not up to you. It's still up to her. But it, according to what you're saying, it is dependent on you giving her the
ring.
diamond ring. So this is the dependent variable. And not the whole thing, but marrying the person. Right? Will marry, the will is the relationship. You must take a stand. You can't say, will she marry me if I give her a diamond ring? Because that's a question. You can't say she married me because I gave her, well, that could still be a hypothesis, but in most cases, the past tense will become the conclusion because it's after you've done the experiment. So can you see what we're doing here, guys? I hope you realize we've learned a few skills here. What is the independent variable? What is the dependent variable? What is the relationship? And what is the hypothesis? So I want you to practice that. Don't practice giving the ring. It's not good because she may clap you and run away with your diamond ring, AB. It's no good. Diamond rings don't come cheap, eh?
Be careful.
Unless you're buying it from those illegal miners there in the other mine there near Benoni. Name the independent and dependent variables in this experiment. And in this experiment, the independent variable, as I said earlier, will be the ultraviolet light because you, the investigator, is in control of that. The dependent variable is the mutation or the change in the bacteria because that you're not in control of. Which apparatus, A or B, was the control and which was the experiment? A was the experiment and B was the control. Let's see why. One petri dish A was placed under an ultraviolet light and the other one was left in the dark. We wanted to know whether ultraviolet rays causes this. So the one that we treated with ultraviolet rays, that becomes the experiment, and the one was just left in the dark. That means it wasn't getting this ultraviolet rays. So the one where we remove that factor what we're testing for. What are we testing for? Whether ultraviolet rays causes a mutation. So where we do not use this ultraviolet ray, that becomes the control. Because what if there was a mutation anyway in the ones that were in the dark? That means it was not the ultraviolet rays that caused the mutation. Do you understand that? The logic behind that? Good. What was the result for the control? All the bacterial colonies were red, and that you're getting from the text, guys. You don't have to be a rocket scientist to know that. It's in the text. Explain any two changes to the method that would improve the reliability. Here's another skill we have to learn today. Good, I'm glad we've added another skill here, reliability. Reliability and validity. Reliability means if we repeat this experiment, will we get the same result? Now, let's take that to real life. AB, if I'm a guy that lies to you every time you ask me, something, I lie to you. I lie to you every time. And James, on the other hand, every time you ask him for something, he gives you the truth. Who's reliable?
It's
for
James.
you.
James.
James, you're going to consider reliable. Why?
Yes.
Because every time he's giving the
Keeps
right
his promises.
answer. And with me, I'm giving you all bullshit all the time. Huh? Okay. So therefore, in the same thing, if the result is the same, then the experiment is reliable. It's like marks. Let's say we give a test. I give you a test, AB, and I give you the test, I mark it, I give you 25 out of 50. John marks it, he gets 26 out of 50. James marks it, he gets 24 out of 50. Is the test fairly reliable?
No.
Why? Because it's a 1 mark variation.
Yes.
It's more or less reliable. But if, go the other way, James got 10 out of 50, John got 40 out of 50, and I got 20. Now it's way out, isn't it? 24, 25, all right, there's a slight variation. It's not too bad, but it's reliable. Here now, there's no reliability there, right? Okay? Are we understanding that? Good. So the first thing you must do to improve reliability, you must repeat the experiment. Why? If you repeat the experiment, you should get the same result. Secondly, the other way of doing it is to increase the sample size, because the more sample you're using, you're getting more results, and therefore, repeatability of the result is evident in that as well. Let's move on. The table below shows the percentage relative risk of a mutation occurring during interphase of meiosis at different ages. So they're giving you a table, and you have this in your notes. In age, they're giving you the age, and on this side, the relative chance of a mutation occurring during interphase. And they give you different dates, less than 20, 21 to 40, and so on here, and they're giving you that. Important thing is here, we need to use the data in the table to draw a histogram. And again, this is also in your notes, so I'm just going to go straight to it. Every graph, remember the 4 graphs that you need to be able to draw: line graph, bar graph, histogram, and a pie chart. So today we're looking at a histogram. The thing about a histogram is ideal for groups of information like this, and notice there are no spaces between the bars. That makes it a histogram. Heading, you must have a caption for all your graphs, every one, including the independent and dependent variable, to get 1 mark. On this side, you must be able to have interval which is equal, and you must have a heading for this with the unit. If you had that and that together, you get another mark. At the bottom, you must have the intervals. Your bars must be of equal width, and if there were bar graph, they would have to be equally spaced also, but here there's no space. But they must all be equal width, okay? And you must have the x-axis name, which is age, in years. Notice the units come here in this part, not in a heading. Then you would have got 1 mark for all of that. And then for plotting, again, plotting is the easy part. You plot your information and try and get into the habit of writing the plotting so the marker can see that you're doing the right thing. Guys, you cannot just write here 7,5, 18, 37,5, 57, and 74,5. Not allowed. You must have a reasonable scale, and these things must fit into those scales. In that, I give to AB.
Thank you so much. There is a flood of questions from the mindsetters, I must say that, Aslam.
Good.
But we'll tackle them after this break. Keep them coming. We're here for you. Otherwise, we'll see you after this break. Welcome back, awesome and beautiful mindsetters. Now we have your questions, and I must urge you that if you've taken the test yourself questions and you are struggling to submit, we have an easy way. Let us know on Facebook, and I'll send you the direct link on where can you submit your answers, because we'd love you to get yourself to that airtime voucher, probably sponsored by Vodacom. And I should say this, Aslam, some mindsetters submit blank answers. So that's not going to take you anywhere, and this is to help you. That is why it's called Test Yourself. So if you're not testing yourself, I don't know.
Guys, you can't shoot blanks at this level. All right, let's move on. Maybe we're just going to take this last question, and then the challenge we're going to answer for them, and then we'll take their questions. Cool?
Cool.
Guys, that multiple choice questions and the one-word answers are absolutely crucial, because they form, more importantly, your Section A type questions. What we are trying to do is we're trying to get you to practice Section A. If you do well in Section A, the rest of the questions can become easier for you as well. Generally, we find a child that does well in Section A does well throughout the paper thereafter. So polish those skills for Section A, multiple choice, multiple matching, one-word answers. Polish those skills and keep posting on the link that is provided. And like AB told you earlier, if you're having a problem, make a comment on the page, and we can then help you with that. Let's move on. An essay question. Every week we're trying to give you one essay per chapter, so it makes your life actually easy to look at essays as well. Organisms display 2 distinct types of cell division, which give rise to the formation of new cells. Discuss the differences between these 2 types of cell division, and state the significance of each. So there's 2 verbs here. One is discuss, and one is state. Cwhatever comes after the verb is what you have to do. Discuss the differences between these 2 types of cell division. What are the 2 types of cell divisions? Mitosis and meiosis. So they want you to talk about the difference, and one of the questions from the learners was the difference between mitosis and meiosis. So please pay attention now. We're not going to take your question later. This is the answer to that question. And also the significance of each type. Good? And how do we tackle this answer? Now, for time, we're going to just put the answer up immediately and just run through it. We're not going to do the whole answer, remember that, because that's not going to do any help for you. What do we do here? We start by saying the differences between mitosis and meiosis, because that's what the question asked us to do. Differences between the 2 types. The mark is not for that heading. The mark is that you have identified mitosis and meiosis as being the 2 types, because the question did not mention the names. By you mentioning the names mitosis and meiosis, you're getting that 1 mark. You may not have put it in a heading. You may have put it further down, but you would have got the mark. Now, The best way to start this thing here is to talk about the early parts. How many times does mitosis? The nucleus divides once, whereas during meiosis, divides twice. Notice, we're not just giving all the mitosis and then all the meiosis. We're discussing them as differences immediately of both of them. So that's the first one. During mitosis, 2 cells produce, and meiosis, 4 cells. Mitosis occurs in somatic cells. Again, another question from a learner. Where in the reproductive cells does this thing take place? It's not in a cell. It takes place in the reproductive organs for the production of sex cells. Okay? In the male, it will be in the testes. In the female, in the ovaries. In plants, you may have different structures as well. In a male flowering plant, you get the anther. Okay? And then in pine, if you watch the grade 11, you'll notice that you have male cones and female cones as well. Cells produced during mitosis are exact copies of the cell that it's producing from, whereas in meiosis, the cells are genetically different. In meiosis 1, homologous chromosomes are arranged in pairs during prophase 1. And in mitosis, are not arranged in pairs during prophase, so the 1 falls away there, right? I'm reading too fast for my own good. And homologous chromosomes occur in pairs during prophase 1 in meiosis. Crossing over takes place in meiosis, and it does not take place in mitosis. In metaphase in mitosis, chromosome lies singly at the equator, whereas metaphase 1 in meiosis, they lie in pairs. During anaphase in mitosis, chromatids pull apart, whereas anaphase of meiosis 1, chromosomes are pulled apart. And the significances, mitosis is ideal for repair, replacement, and growth. Meiosis maintains chromosome number, is essential for production of sex cells, introduces variety in species, allows for alternation in genes. Now notice, guys, I've rushed through that part. I do agree. But it's there. The answer is there. You can look at it, answer in your own time. What's important for me is I want you to look at the arrangement. Look at the arrangement of the question. It says, discuss the difference between 2 types of cell division and state the significance, okay? So let's look at how we answer this question. We first looked at the differences, yes? We dealt with the differences, and then we did it. So whatever comes first in that question, you deal with that first, and then. If you can deal with the significance, like last year they gave a question in paper 1 in evolution, where the significance could come in with the difference. For example, humans have larger brains than other organisms. The significance of this is, so the difference and the significance tied together, then you can do it in 1 unit. But here, it's totally different, and you can do it. So you're getting a mark for logic. Again, remember, you're getting a mark for relevance, logic, and comprehensiveness. 1 mark for relevance. If you wrote anything else besides what we asked here, you're going to lose that mark. If it didn't follow logic, you started here, then you went there, then you went there, then you went there, 1 significance, 1 this, 1 that, you're going to lose that mark. And if you did not answer the question, which were the 2 parts of this question? 1 was the differences between mitosis and meiosis, and 1 was the significance. So if you left out 1 of those 2 things, you wouldn't have got the mark for comprehensive. Remember, these are the marks for synthesis. 3 marks. Good. Let's look at the challenge question. If X units are present in a nucleus of a cell which has just divided, what is the relative amount? Again, we don't have to read the question. You've done that already. And obvious, because it's mitosis and it's exact duplication, the answer has to be X, the same amount of chromosomes, because this is mitosis. Okay? Now this one here. In a rhino consists of 82, how many chromosomes would it have? In the cells of its ears. The cells of its ears would be somatic cells. Let me write that down. Somatic cells. And the somatic cell would have diploid number. And the sex cells, or gametes, should have N. Haploid. Diploid, let me write that so you know what I'm talking about. This now it's Greek to you. Haploid. Haploid is N, diploid is 2N. Full set of chromosomes, 2N. Half, haploid, N. So in the cell of the ears, somatic cells, so therefore obviously it should be 2N, and therefore it should be 82. In the cells of its horn, also somatic cells, therefore it should be also 82. In the sperm cell, a sperm cell is a sex cell, haploid, divide by 2 should be 41. In the cells during anaphase 2 of meiosis, Anaphase, 2. Remember, in the first half of meiosis, the chromosomes move to opposite poles already. So in the first division of meiosis, the chromosome number is already half. So in anaphase 2, it should be half already, 41. In the cells during prophase 1 of meiosis, the cell chromosome has not divided yet. So therefore, it should be still 82. I'm hoping we got through that. Abi, now it's your show.
All right, let's start from...
Let me extend that page.
Let's start here.
Start from the beginning.
Yeah.
Always nice to start at the start.
Okay, unfortunately, I'm not going to start. Okay, let's
No,
start.
anyway.
Let's start in the beginning. Here's Edson saying, hi, my sisters. It's my birthday today.
Happy birthday.
Happy birthday, Edson Bayiza. I hope you're not biasing. His question says, why do Down syndrome happens at chromosomes number 21 only?
Okay, Down syndrome, you see, it all depends what happens with which chromosome. There are thousands of other syndromes that happen. But if it is that chromosome, because that chromosome controls that particular, remember, we spoke of the whole human genome. What scientists have done is they can tell you which chromosome in your body, what gene is found in each locus. Locus is the position of a gene on a chromosome. They can tell you that. And they can tell you exactly which chromosome deals with what. Now, chromosome number 21, if there are 3 instead of 2 in an individual, he or she suffers from Down syndrome. But let's extend that question to talk about this. Somebody else asked about non-disjunction, so I'm going to combine the two. Let's look at this term called non-disjunction. This means during anaphase, it can be 1 or 2. The chromosomes or the chromatids separate. One chromosome separate. In anaphase 2, the chromatids separate. If the chromosomes do not separate during anaphase 1 or anaphase 2, then the resultant cell, one of the resultant cells, let's say we had a cell with 2 chromosomes. And during anaphase 1, or no, 2 is too few to show there, right? Let's say it had 4 chromosomes. During anaphase, now what happens is that these 2 did not separate. So in other words, when anaphase is taking place, these 2 stayed together, plus the 1 separated one came here. And on this side, we're only getting 1, because 3 went that way, 1 remained on the other side. So in other words, when this cell now forms, this cell is going to have 3 of this, and the other cell is only going to have 1. We call this aneuploidy. That means the cell is having 1 extra chromosome here. Okay? In Down syndrome, this chromosome that we are talking about is chromosome number 21. Okay? If this is a gamete, let's say this is an egg cell. It has this extra 21. And we have a sperm, nice little spermie there, coming to this egg that has 24 chromosomes. Remember? An egg supposed to have 23, but because we have 1 extra, it will have 24. The sperm, on the other hand, is normal. It's got 23. 23 plus the 24 will give you 47 chromosomes. If it's on 21, it is Down's syndrome. That's the answer for that one.
Talking about dysfunction, here's another question following that up. Can dysfunction occur in the last phase of meiosis?
Last phase, telophase? No. Disjunction or dysfunction.
Disjunction.
Disjunction can only occur in one of the anaphases. Although, for your studies, you can just say in meiosis 1 or meiosis 2. But generally, remember guys, Abraham also, that the bottom line is, we have just given names to phases. In reality, the cell doesn't say, now it is prophase, I need to do this. Okay, now prophase is finished, it's meta. No, it happens in a twitch. We have just divided it so that we can understand it. Okay?
Okay.
Other questions?
Here's another one. I think you'll enjoy this one. Where is it now? Okay.
How do you
From
know what I'm
Mandy,
going to enjoy?
You are going to enjoy this one. What are the consequences of abnormal meiosis?
What are the consequences of abnormal meiosis? There's your answer. I don't have to do it again. This is abnormal meiosis. Abnormal meiosis means something goes wrong. And for our syllabus, the only one that we have to worry about is the one I've described now, Down syndrome.
Okay. When does random assortment take place, and how
Good
does
question.
it differ
I saw that
from
earlier.
crossing over?
Random assortment. Do not confuse this with independent assortment, which we will talk about in genetics. A random assortment, remember we said that in metaphase, the homologous chromosomes are lying next to each other at the equator. Let's say these are homologous chromosomes. They are assorted in this way, AB. They could be assorted in this way, or we can take these guys and put them on that side, and take these guys and put them on this side. Or we can leave them the way they are and swap them. This one goes at the bottom, that one goes on the top. In other words, random means, guys, there's no guarantee in which position which chromosome is going to be positioned at the equator. So when they separate, there's no guarantee which chromosome is going to go to which sperm cell or egg cell. And because of this, this also brings about variety. So two things bring about variety in meiosis. One is crossing over during meiosis prophase 1 and a random assortment. It's like the lotto. If you knew which balls were coming out on tonight or Saturday, would you be sitting here next to me, AB?
No.
Not at all. That's why, because it is random. Random means there's no guarantee. Next.
Obviously.
Obvious.
Right. Here's another one that you may think it's obvious. Mpaga Lerato, shouldn't crossing over occur before meiosis to
Crossing
occur?
over has to obviously happen in prophase 1. Notice we're saying crossing over takes place during prophase 1. It can't take place after that, okay? Because the homologous chromosomes are not lying next to each other. Anymore?
Okay. I still don't understand the phases. Could the teacher write them for us?
The phases, very quickly, we can write for you, yes. One is, hey, what happened to that color? One is interphase. The interphase is actually a phase between two phases. The old textbooks will say it's a resting phase, and some of the new textbooks. But an important process takes place there, and that is DNA replication. Then comes prophase, the first phase, I-P, prophase. And we spoke what happens there, crossing over and the rest of it. Prophase 1. Then we have metaphase. Metaphase middle. Then we have ana. And then we have telophase. Remember in meiosis, it's 1, 1, 1. Prophase 1, metaphase 1, anaphase 1, telophase 1, and 2, 2, 2, 2. A, B.
That's where we end it today. Thank you for all the suggestions that you've also submitted. We'll choose next week. Keep your suggestions coming on what should we call our Wednesday. But for today, we just want to say thank you very much for your time. Keep learning, and all the best to everybody that is writing their sub-exams. We wish you nothing else but the best. But today, the A Squared just want to say we leave you in peace. Keep on believing in yourself.
We'll use your name next week, Wednesday.
Obvious.
Obvious.